This video is going to talk about the processes and methods used to analyze the genetic variation of the samples found by the SNP assays. The program that we use is called Genome Studio. The basic process of calling SNPs using this software is built on a 6-step workflow process. The first step is to load the project. This is done by importing all of the data from the assays. Next, the experiment's quality needs to be determined. Looking in the control dashboard, you can see the different steps of the assay and notice erroneous data that is present in the samples. This data can be taken out of the clusters later. After this, all of the samples need to be sorted due to the inaccuracy of some samples. In this graph, you can see that some of the samples here have a lower call rate. Here you can see what the total call rate of the project is defined as. The second plot shows the GC as a function of call rate. The GC, or gen call score, is a parameter that is used to ensure accuracy for the genotype. GC score typically lowers as a sample is further from a cluster. The next step in the workflow process is reclustering. In this plot, you can see how the reclustering failed because a bunch of the data points are together and you cannot make distinct groups. In the next plot, you can see that reclustering was successful because there were 3 distinct clusters. There are 3 ways of clustering the data. The first is to cluster all the SNPs, and this requires extreme caution. The next is to cluster a subset of SNPs, which uses a filter to identify problematic loci, and then the third is to skip reclustering, and this uses a standard cluster file. The next step in the process is locus analysis. Here you can see a successful SNP locus due to the 3 distinct allele frequency groups. During this step, you can also look at the Y chromosome SNPs. You should remember that Y chromosome SNPs have 1 copy and should result in a homozygote genotype call. The last step of the process is to save and export the SNPs. The position of the clusters can be saved. Thanks so much for watching.